Hi, my name is Colby Watase.
And I'm Jayna Wong. Our project is Effective Alternative Ribosomal Proteins on Ribosome Localization in Mycobacterium smegmatis.
Tuberculosis has been and still is ranked top cause of death by infectious disease. According to the World Health Organization's 2019 Global Tuberculosis Report, there was an estimated TB incidence of 10 million and an estimated 1.2 million deaths in 2018. The causative agent of tuberculosis is Mycobacterium tuberculosis, abbreviated as MTB. During MTB infection, structures called granulomas form at sites of infection. The figure to the right represents a granuloma consisting of immune cells and MTB. MTB can encounter zinc-limited conditions within granulomas. Despite zinc-limited conditions, MTB is able to survive within the host. MTB expresses what are referred to as alternative ribosomal proteins in zinc-limited conditions, indicating possible involvement in survival. Many bacteria have genes encoding alternative ribosomal proteins, abbreviated as AltRPs, which are paralogs of primary ribosomal proteins, abbreviated as PrimRPs. PrimRPs and AltRPs differ in binding capabilities for the zinc ion, an essential cofactor for many biological processes. S18-2 is an AltRP that does not bind zinc and therefore does not require it to function. As depicted in the top figure, the S18-2 AltRP is encoded by the RPSR2 gene on the AltRP operon that is negatively regulated by zinc. As depicted in the figure on the right, during AltRP expression in low zinc conditions, some ribosomes may incorporate AltRPs instead of PrimRPs during assembly, yielding these alternative ribosomes. Assembly of alternative ribosomes contributes to varied relative abundances of different ribosomes within a cell, possibly changing protein synthesis efficiency.
Mycobacterium smegmatis is used as a non-pathogenic and fast-growing model of MTB. Morphological changes were observed in M. smegmatis that expressed AltRPs in zinc-limited conditions. In the absence of zinc, wild-type smegmatis cells elongated, and the AltRP mutant, which lacks the AltRP operon, did not, and it resembled cells grown in zinc-replete conditions. Furthermore, the presence of zinc alters distribution of polyphosphate bodies. Wild-type smegmatis grown in zinc-replete media have polar phosphate bodies, while wild-type smegmatis grown in zinc-limited media have multiple polyphosphate bodies spaced regularly throughout the elongated cell. Growth in zinc-limited media also causes the nucleoid to condense. These morphological changes during zinc depletion indicated the possibility of AltRPs having function beyond purely replacing PrimRPs in ribosomes. Previous studies have shown that distribution of ribosomes in E. coli varies with growth phase and conditions. There are AltRP-dependent translational differences in smegmatis that reflect the morphological differences between PrimRP and AltRP expressing smegmatis. Differential localization of ribosomes may change access to mRNA pools and ribosome-associated proteins, affecting the mRNAs translated and the proteins synthesized. Condensation of DNA alters the pool of mRNA that is transcribed. Therefore, differential localization may dictate which proteins are synthesized. Due to these reasons, organizational differences of ribosomes are expected in M. smegmatis grown in zinc-replete and zinc-limited conditions.
We hypothesize that ribosomes are localized differently depending on the incorporation of PrimRPs or AltRPs during ribosome assembly in zinc-limited conditions. Our objective was to clone a turbo-YFP tag at the N or C terminus of PrimRP S18-1 encoded by RPSR1 and AltRP S18-2 encoded by RPSR2. Additionally, we cloned a turbo-YFP tag at the N or C terminus of core ribosomal protein S2 present in both primary and alternative ribosomes. The fluorescent tag allows visualization by fluorescence microscopy. Our research has a variety of possible applications, including understanding survival mechanisms of MTB in zinc-limited conditions and identifying potential targets for anti-TB treatment.
Primers were designed in order to add enzyme restriction sites to the 3' and 5' ends of the construct, as well as a flexible linker region between the ribosomal protein and the YFP tag. The flexible linker allows independent folding of each protein. Enzyme restriction sites were added in order to facilitate insertion into the E. coli mycobacterial shuttle plasmid pNet1. The first PCR reaction seen on the top amplified the target gene sequences with these primers. Generating the fusion protein gene by overlap extension PCR involved 2 steps. The first PCR hybridizes the ribosomal protein and YFP genes with the overlapping sequences of the linker. The second PCR uses the forward and reverse primers of what would be the full gene product in order to amplify one continuous gene. The fusion protein construct was then inserted into peanut-1 using restriction enzyme digest and ligation procedures. Restriction enzymes cut DNA at specific sequences, usually forming single-stranded sticky ends that can then pair with complementary sequences. The fusion protein genes with RPSR1 or RPSR2 contained EcoR1 and NDE1 restriction enzyme recognition sequences on the 3' and 5' ends. The peanut plasmid also has these sites, which are highlighted in yellow in the figure on the left. The construct with RPSB contained EcoR5 rather than EcoR1 due to the presence of an EcoR1 recognition sequence within the RPSB sequence. Each fusion protein gene and the peanut-1 plasmid was digested with the 2 restriction enzymes, generating complementary ends. Each digested construct was incubated with the digested peanut-1 plasmid and T4 DNA ligase, resulting in ligation of the constructs into the plasmid. In order to amplify the plasmid, peanut was transformed into competent E. coli using the heat shock method. Transformed cells were spread and grown on LB plates with kanamycin in order to select for cells that successfully took up peanut-1, which confers resistance to kanamycin. Colony PCR was used to screen for colonies with both antibiotic resistance and fusion protein construct. These images show the PCR products run on agarose gels. They confirm that the inserts have the correct size, indicating that ligation and transformation were successful. Successful insertion of the constructs was confirmed by sequencing. Each peanut-1 plasmid containing a different fusion protein gene was then transformed into M. smegmatis using established Prisic lab electroporation protocol.
Wild-type and M-cherry smegmatis were grown in parallel with transformed smegmatis. M-cherry is a reported strain for AltRP expression. Smegmatis from frozen glycerol stalks were inoculated in liquid cultures of 7H9 ADC with kanamycin and grown in a 37-degree shaking incubator for 18 hours. After incubation, the cells were centrifuged, washed, and resuspended in Sutton's medium with kanamycin. Cultures were normalized to OD1 as to achieve a theoretical starting OD of 0.001 in 50 milliliters of Sutton's medium. Each of these cultures were grown in Sutton's medium. Wild type was grown in both zinc-limited and zinc-replete conditions. OD600 was measured over 3 days in 24-hour increments. After 48 hours, caprolactam was added to induce fluorescence under the nitrile-inducible promoter of peanut-1. To optimize the expression of fluorescence for fluorescence microscopy, cells were centrifuged and resuspended in fresh media after 3 hours of exposure to the inducer. Cells were allowed to grow for 24 additional hours to stationary phase. Depicted here are the growth curves of our smegmatis strains over the 72 hours. The results essentially indicated that growth was not significantly impacted by synthesis of the fusion proteins. After 72 hours of growth, YFP fluorescence was measured at a 520 nanometer excitation and a 550 nanometer emission. The peanut YFP smegmatis control provided a reference for fluorescence. Depicted here is a graph of fluorescence for tagged AltRP S18-2 encoded by RPSR2. The N-terminus tagged smegmatis showed essentially no fluorescence, while the fluorescence of the C-terminus tagged smegmatis was 34% relative to the YFP control. The lack of fluorescence observed in the N-terminus tagged smegmatis may indicate that the orientation of YFP tagged on the N-terminus tag of AltRP S18-2 interferes with proper folding of the individual proteins. Depicted on the right, there was a mixed population of elongated and shorter N-terminus tagged AltRP S18-2 cells in stationary phase. In comparison, there were consistently elongated C-terminus tagged AltRP S18-2 cells, as depicted on the left. The results indicated that the N-terminus tag may disrupt the function of RPSR2.
Because cellular morphology was altered between the two AltRP S18-2 constructs, we wanted to observe if colony morphology was also impacted. After 48 hours of growth in Sutton's, cells were spread onto Sutton's plates with 28 millimolar caprolactam and without supplemented zinc. The N-terminus tagged S18-2 strain, seen on the far right, has markedly different colony morphology than the C-terminus tagged strain and wild-type smegmatis on the left and middle. N-terminus tagged colonies are elevated and wrinkled throughout, while C-terminus tagged strains and wild-type smegmatis have concentrated regions of elevation but are smooth and flat otherwise. This indicates that AltRPs could possibly play a role in modification of the cell wall and related processes. All other constructs will be plated in the future. Ribosomes were isolated using anion exchange columns to determine if fusion proteins were being incorporated into ribosomes. In short, M. smegmatis cells were centrifuged, resuspended in an ion buffer, and lysed. Lysate was applied to the columns, ion buffers of increasing concentration were run through, and the flow-through was collected. Flow-through fractions were analyzed for fluorescence. Fluorescence of the fractions preliminarily indicates that the C-terminus tagged fusion protein, seen in dark blue, is being incorporated into the ribosome. Ribosomes elute in the Q2 fraction where fluorescence is present. Fluorescence of C-terminus tagged fusion proteins in the Q1 fraction is likely protein that is not incorporated into ribosomes. Western blot will be performed on the ribosome isolation fractions with anti-turbo YFP antibodies in order to confirm incorporation of the fusion proteins into the ribosomes. Pictured here are images of YFP and C-terminus tagged S18-2 using fluorescence microscopy. Fluorescence in YFP M. smegmatis is diffuse, while fluorescence in the tagged strain is clearly localized into puncta. This also indicates that the fusion protein constructs are incorporating into ribosomes. The appearance of these puncta in regularly spaced intervals possibly indicates that the ribosomes are localizing around polyphosphate bodies. This is a graph of fluorescence of tagged prime-RP S18-1, which is encoded by RPSR1. There is essentially no fluorescence in neither the N-terminus nor C-terminus tagged S18-1. This lack of fluorescence may be because these cells were grown in zinc-limited conditions when AltRPs are incorporated into ribosomes rather than prime-RPs. For future trials, these will be grown in zinc-replete conditions in order to observe localization of primary ribosomes.
The graph of fluorescence of C-terminus tagged ribosomal protein S2 encoded by RPSB is shown here. As you recall, S2 is present in both primary and alternative ribosomes, so it was grown in both zinc-replete and zinc-limited conditions. Additionally, uninduced controls were grown. C-terminus tagged S2 showed expression of YFP in both zinc-replete and zinc-limited conditions without exposure to caprolactam, indicating leaky expression of the plasmid in those strains. Previous studies have shown a basal level of pNet expression without induction, so future experiments will evaluate basal level expression in other strains. But, as established previously, leaky expression does not affect growth. Depicted above are really nice pictures we took using fluorescence microscopy. On the left, we see our YFP control with uniform distribution of fluorescence. As with C-terminus tagged AltRP S18-2, the C-terminus tagged S2 ribosomes are localizing into puncta that are in regular intervals throughout the cell and at the poles. Due to the short length of the cells grown in zinc-limited conditions, it does not appear that the cells had reached a stationary phase. Further imaging will be performed after a longer incubation period. Overall, our results that indicate ribosome localization is affected support the hypothesis that ribosomes are localized differently in zinc-replete and zinc-limited conditions. Due to the recent circumstances, our lab research time, unfortunately, was limited. However, we have planned the next steps of our study. We will be growing and visualizing N-terminus tagged S2 and the N and C-terminus tagged prime-RP S18-1s. We have also transformed all constructs into delta AltRP mutants that lack the AltRP operon in order to compare cellular morphology, colony morphology, and ribosome localization using cells that lack the native AltRPs. We plan on purifying ribosomes from all strains and performing Western blots using anti-turbo YFP antibodies to observe whether or not incorporation into the ribosome occurred. Lastly, we will perform super-resolution microscopy with Dr. Nicholas James to observe ribosome localization in greater detail.
Mahalo to Dr. Sladjana Prisic, our faculty mentor, Endrei Marcantonio, our PhD student mentor, and Dr. Nicholas James, who helped with fluorescence microscopy. We would also like to acknowledge Alexa and Brandy, as well as the rest of Prisic's group, for their ongoing support. And finally, we would like to thank the University at Hawaii at Manoa's Undergraduate Research Opportunities Program, which allowed us to do this project.